Yes. So I'm going to record here our lecture, or the second part of the lecture on the heart. So I'm going to turn my video off, pull the screen up here big. Okay, so picking up where the last video left off, we finished talking about kind of the anatomical stuff associated with the heart. So we kind of have an idea of how the heart's built, which is going to help us the rest of the way for understanding how the heart functions. Okay, so we're ready to talk about, if my computer will cooperate, there we go. How does blood move? And in parentheses there, you can see it says part 1. Part 2 will be discussed in our next lecture, where we include blood vessels, where blood also moves. We've talked a little bit about this, thinking back to our last lecture, where we talked about the flow of lymph, kind of discussed how fluids flow in our body, which relates to kind of a newer concept called pressure gradients. Thinking back to the very beginning of the semester, we talked about concentration gradients, where, like oxygen moves from high concentration to low concentration. The same kind of concept exists here with the flow of blood, like it did with the flow of lymph in our last lecture. It's all about high pressure to low pressure. And this is actually a law that goes back to Isaac Newton. Newton's law of motion, or laws of motion applied to fluids, states that a fluid does not move when pressure is the same throughout. We show two kind of examples. We could pretend that like this is a left ventricle, this is the aorta. P sub 1 represents pressure 1, and in this case the left ventricle, 80 mmHg. mmHg represents millimeters of mercury, one of the kind of standard units of measurements using of pressure in our body. And then P2, pressure 2, this would be in the aorta, is also 80 millimeters of mercury. Since those are equal, there is no gradient, so flow is not going to occur. What our heart does is it contracts and squeezes, which is evident back on this page. When the ventricles, for example, squeeze, pressure inside goes up, which you can see here. P sub 1 now has risen to 100 millimeters of mercury. P sub 2 hasn't changed. There is a pressure gradient, and it states here a fluid is only going to flow when pressure is higher in one area than another. Flows from high pressure to lower area, or lower pressure area. So it would go from P1 to P2, or out of the left ventricle into the aorta. Now, remembering back to our lecture on muscles, we kind of introduced a little bit of information on the 3 types. We focused the most on skeletal muscle in that lecture, but we did discuss smooth muscle and cardiac muscle. Sorry, it's kind of cut off here just because of the stuff here at the top of my screen, but you can kind of read through it anyway, and you guys have the text on your paper anyway. So remember, cardiac muscle is striated. So I can actually read it. It's striated like skeletal muscle, but has branches giving individual cells a stair-step appearance. Here's what some of those cardiac muscle fibers look like. One, you can see right here is not branched, but this one next door is branched, connected to another branched one. The fancy term for these muscle tissue cells are cardiac myocytes, or just cardiac muscle fibers. And they're joined end to end by thickened connections of the sarcolemma, what we call intercalated discs, which was something that we saw in that picture of cardiac muscle under a microscope, and I kind of mentioned at the time. We would talk about it again, and here we are talking about it again. Now, 3 features I want to talk about with these intercalated discs: these little finger-like extensions that basically enable the cells to interlock one another. Those are called interdigitating folds. So it increases surface area and increases the amount of intercellular contact. So it's like a jigsaw puzzle holding the cells together. So it's a really nice, strong connection. And then also increasing the strength of connection between them are, I always kind of think of them like welds that you performed to connect two pieces of metal together. These things called desmosomes. They're a form of cellular junction that is going to really tightly join those cells together, so that when the myocytes contract and pull, they work as a team instead of pulling apart. Kind of physiologically, the most important ones that we're going to talk about are a component of our intercalated disc, what we call gap junctions. They're basically just channels that allow the ions to flow from one cell to another. And in doing so, this allows action potentials to thus spread from cell to cell. Thinking back to both when we've talked about skeletal muscle action potentials and neuron action potentials, when things like sodium flowed into a cell, it changed its resting membrane potential enough that, if it reached threshold, it created an action potential. The same thing takes place in cardiac muscle fibers as it does in neurons and skeletal muscle fibers. And so when these ions flow from one cell to another, the cells all get kind of excited at the same time. Okay? And that's really important because it's going to allow for the muscle fibers to then contract together. Because just like we learned with skeletal muscles, when cardiac muscle fibers get excited, they contract. And this is something super important because it's going to help control and direct the flow of blood in a very coordinated manner. Because what we want is not the entire heart contracting at one time, but, and we kind of talked about this on this slide, we want both atria chambers to contract, pushing blood into the ventricles. While that's taking place, the ventricles must be relaxed so that their pressure is low. And then once that blood fills up the ventricles, we need the atria to relax, and then both ventricles to contract to eject blood out of the heart. So it's a very coordinated series of events, and gap junctions help with this. Gap junctions allow for the nerve signal to go from cell to cell, but where does that nerve signal originally come from? Well, let's talk about that on this slide. So, I pulled it off here so you could see that top term in case you needed to fill it in. Top term is autorhythmic, which we've introduced before back in that lecture on muscles. So reading through, about 1% of cardiac myocytes are described as being autorhythmic, meaning they depolarize spontaneously at regular time intervals. During development, some of these myocytes become specialized for generating action potential, and these are the cells that essentially become the wiring of your heart, what we call the cardiac conduction system, that not only controls the route, but the timing of electrical conduction in our heart. It all begins with a structure called the SA node, short for sinoatrial node, which is found up here in the right atrium. And your SA node is commonly called your pacemaker. And it creates the original signal that causes each heartbeat and therefore determines heart rate by creating what's called a pacemaker potential, which is just simply where it depolarizes to threshold and then fires an action potential, shown here on the bottom right of the page. When the SA node fires an action potential, that action potential spreads not only through the right atrium, but across through the inner atrial septum and then causes the left atrium to fire as well. And then following that, both the atria contract in unison. So very, very cool. The way that those action potentials are able to spread away from that SA node are through those gap junctions that we just talked about. Now, by conducting along the atrial muscle fibers, an action potential will reach the second node, what we call the AV node or atrioventricular node, which is found in that interatrial septum, which is shown in our picture right here. Notice the two are not directly connected, whereas everything else is kind of wired together. The AV node is where that action potential begins to travel down into the ventricles. The AV node allows for the signal to really kind of slow down. We call this an AV node delay, which provides time for the atria to empty their blood into the ventricles. Otherwise the ventricles will be contracting before they're full, and there'd be no point in that. From the AV node, the action potential enters the AV bundle, or bundle of His. Atrioventricular bundle is the only site where that action potential can travel from the atria into the ventricles. And it's because of the fibrous skeleton of the heart, which we don't really get into the fibrous skeleton in this class, but that's where it has to go. After propagating along that AV bundle, we then see a fork come in the road, forming what's called a right and left bundle branch that carry that nerve signal all the way down to the apex, the point of the heart, through the interventricular septum, that wall between the two ventricles. And then finally, we use the Purkinje fibers to rapidly conduct the action potential from the apex upward to the remainder parts of the ventricular myocardium. And that's what causes the ventricles to contract. So that's what these fibers are throughout our two ventricles. Following that, blood is going to be squeezed up and towards the semilunar valves, the aortic valve, the pulmonary valve, to allow blood to leave the right ventricle and the left ventricle. Now, if we left the SA node alone, it would set a heart rate that never changed. Okay? It would be a constant beat that never, goes up or down. But obviously, we've talked about facts that, the fact that, your sympathetic nervous system can speed up your heart rate, your parasympathetic can slow it down. We've talked about various hormones like epinephrine. The original signal is always going to originate in the SA node, but nerve impulses from our autonomic nervous system, either sympathetic or parasympathetic, and blood-borne hormones like epinephrine will modify the timing because it can either tell the pacemaker to fire more often or less often. Okay? We can also modify not only the timing, but the strength of the heartbeat, what we call contractility. But once again, they do not establish the fundamental rhythm. Okay, let's talk about action potentials some more and contraction of our cardiac muscle fibers. As you can see here, this is an illustration showing you a action potential in a cardiac muscle fiber, cardiac myocyte, and it's very different than what we saw both in our neurons and our skeletal muscle fibers. And we have kind of three phases represented here in our figure. Number one phase, two phase, and then the third phase. First phase is called a depolarization phase, or as it's listed here as a rapid depolarization. Where we start is actually quite a bit more negative than we talked about with neurons. Remember, neurons had a resting membrane potential of around -70. We see that cardiac muscle fibers have their resting membrane potential closer to like -90, but the slope upwards is quite a bit more steep. It's not as drawn out, so we depolarize a lot quicker. And it's still the same thing. We still have sodium inflow, but the key is these are called voltage-gated fast sodium channels. And that's because they just open really rapidly and allow for really, really fast flow of the sodium in. Now, unlike what took place in neurons and skeletal muscle fibers, we don't pretty much return right back to resting membrane potential immediately. What we do is we hold on and stay depolarized above zero because of what's called the plateau phase, which is, as it says, the depolarization is maintained because of, this is a new type, oops, sorry, I didn't mean to hit the next button, due to calcium inflow through voltage-gated slow calcium channels and potassium outflow through voltage-gated potassium channels. Now, the voltage-gated potassium channels are the same ones that we saw back when we talked about neurons that would cause the resting membrane, or cause the neuron to return back to resting membrane potential. But the fact is now we're allowing calcium to come in, which kind of negates the flow of potassium out. If you have positive coming in, positive going out, there's not going to be much change, and that's why we see it kind of just staying put where it's at before we hit the repolarization phase, when those calcium channels close and the potassium can continue to outflow, and we return back to resting membrane potential. Now, the whole purpose of having this plateau phase will be discussed on the next page, but it's illustrated here with the stuff down at the very bottom. What we end up having is a really drawn out refractory period. Remember, a refractory period when we talked about neurons was like the point, I use an analogy of like jumping in the air. You can't jump a second time until you land the first time. A really long refractory period in our cardiac myocytes allows for a contraction to kind of last for a while, which is important, and we'll see why here in just a second. So let's talk about that here on this next page. Okay, cardiac muscle fibers, like neurons and skeletal muscle fibers, have a refractory period of time after an action potential begins when an excitable cell temporarily loses its excitability. Okay. Here we show in blue our illustration of the action potential, what we just saw on the previous page. What we're showing here in red is the tension that's developing, so the contraction process that's taking place as we go through that action potential. The time gets expanded out quite a bit, so we kind of have a long contraction period before we relax. Okay, so let's read through why that's important. So in contractile cardiac muscle fibers, the refractory period is long, about 250 milliseconds, due to the prolonged plateau phase of the action potential. Refractory period of these fibers lasts almost as long as the duration of the contraction, about 300 milliseconds. As a result, a contractile cardiac muscle fiber cannot become re-excited until its previous contraction is over, or almost over anyway. Kind of like what we talked about, you can't jump a second time until you've landed the first time. For this reason, summation of contractions and tetanus does not occur in cardiac muscle fibers. Thinking back to when we talked about skeletal muscle, we talked about summation and how, if you hit it with an action potential before the first one is over, skeletal muscle fibers are going to contract stronger until eventually you hit them with so fast of action potentials that they reach what we call tetany, where that muscular contraction is sustained, like what happens when you hold, something from an outstretched arm. You're continuously contracting your skeletal muscles. We cannot continuously contract our heart. It cannot stay contracted, which is important because if your heart stays contracted, it's not going to refill with blood. And the whole reason your heart is contracting is to pump blood. So what good would it be if it was squeezed at all the time and would never refill back up? Obviously, it wouldn't do any good. So all of the stuff I've talked about on the last 2 pages kind of boils down to allowing your heart to refill with blood. Okay. Let's talk about ECGs. All right. Each action potential that travels through our heart can be detected and amplified by what's called an electrocardiograph, which is a machine that, it's a machine that creates what's called an electrocardiogram. An ECG, also sometimes you'll see it listed as an EKG, is what you see in movies when you look up at the monitor and it's going beep, beep, and you see the things going up and down, and then somebody's in the process of dying, it flatlines, and what they are looking at is an ECG. Now there's several components to an ECG. One of them is labeled here on this illustration. The first detectable wave or signal that can be determined is what we call the P wave, a small upward inflection representing the point when the atria are depolarizing. And as you can see at the kind of top of P, the atria begin contracting and will contract to push that blood down into the ventricles. Now, from the beginning of P to the next very prominent segment of a ECG is what we call the PQ segment or PQ interval, as it's listed over here, the time between the P wave and the next wave, and it represents the time required for impulses to travel from our SA node to our AV node. Okay. The QRS complex is the biggest change to the ECG. It involves a small downward Q, very high jumping R, and then a big drop back down to S. Okay, and that's where the QRS complex comes from, where the signal is being produced, or the signal that's being produced in this is due to the action potential traveling from the AV node and all the way down through the ventricular myocardium, causing ventricular depolarization. And notice here at basically the top at R, we begin contracting the ventricles, which we say here is called ventricular systole, which we've seen that word systole already here in this lecture, and I'll get into that here more in just a couple slides. Down here at, well, let's talk about the ST segment next. Okay, the ST segment represents the beginning or the time during which the ventricles contract and are ejecting blood. And then we hit what we call the T wave. T wave is a little bit bigger wave than the P wave. It's a big upward, not big, but it's a fairly big dome-shaped inflection or deflection when the ventricles are repolarizing, returning back to their resting membrane potential so that they can get ready to contract again. Okay. From the beginning of Q all the way down to the end there at T, this is what we call a QT interval, or you'll see it sometimes listed as a QT segment. Represents the beginning of ventricular depolarization to the end of ventricular repolarization. Now, if this segment or this whole picture was bigger, we could have another series of events where a P would start over, somewhere out in here, and then we'd see the same thing with the QRS and then a T, and this continues on and on and on. As far as the exam goes, I will definitely want you to know, what the P wave is, what the T wave is, all those different waves and segments, intervals. You should also be able to just kind of identify them in this illustration. So what I could do is, put a picture of a ECG on there. I might highlight like the T wave and I say what's taking place here. Now, these are pretty standard tests that are performed on people when they go to the hospital, especially if they are, in a state of, heart issue, because there's a lot of information that can be gained from a ECG. I'll kind of show you some examples of irregular ECGs here on this page. I took a class when I was in college. We spent a quarter of the semester just reading ECGs, learning about what first degree AV block meant, what AFib means, what VTAC means, what VFib means, which are all illustrated on this page. Not something I'm going to ask you to know, but just know that there's certain professions that reading these is kind of what they do, especially for people that are performing rehab on individuals that have had a heart attack or some type of cardiac episode. Going back to this illustration is from your textbook. Kind of nice because it's showing you the different steps of the ECG, but also kind of highlighting what's taking place with each section of the heart. So we show here at the top here in our first picture, the P waves beginning because the SA node is fired, and we are creating that action potential not only throughout the right atrium, but it's crossing over into the left atrium. And then following that, you can see the atria are squeezing down, causing contraction, what we call atrial systole, pushing the blood down and into the ventricles. While that's taking place, we carry that signal down into the ventricles, where now the ventricles, those muscle cells are depolarizing. We see that with the QRS complex taking place. Following that, from the S to the T, the ventricles are going through contraction, what we call ventricular systole, pushing the blood up and out of the ventricles into the aorta and into the pulmonary trunk. Following that, the T wave begins as the ventricles start to repolarize, so that this can all begin again. Now you might be wondering, why don't we see anything for atrial repolarization? Because the atrium must return back to their resting membrane potential before they can contract again. Well, atrial repolarization does take place after atrial systole, but the fact that we have this big QRS complex taking place just kind of masks, kind of covers up the electrical signal, so it's not a detectable wave that can be read with a ECG or read on an ECG. Following ventricular repolarization, we're back at baseline, and in ventricular diastole or relaxation, ready for everything to start back over. Now, what I've been using on that page are terms like systole and diastole, which are very important components of what we call the cardiac cycle. Okay, so reading through. As you've learned, the atria and the ventricles depolarize at different times because the conduction system routes cardiac action potentials along a specific pathway. Our cardiac cycle refers to the steps involved in one complete heartbeat and consists of two parts, what we call systole and diastole. Systole is a phase of contraction, which can be both in an atrium or both atria at the same time, what we call atrial systole, and in the ventricle is what we call ventricular systole. Diastole, however, is the opposite of that. It's relaxation or expansion. And we have what we call atrial systole, or diastole, excuse me, when the atria are relaxed, and then ventricular diastole when the ventricles are relaxed. Now, going back to the page here where we discussed heart valves, I had in parentheses ventricular contraction or systole, ventricular relaxation or diastole. And as I walked through that section of the lecture, I talked about the terms systolic, diastolic. Systolic, diastolic refer to measurements of blood pressure, where you go to the doctor, they put the BP cuff on your arm, they give you two numbers, a big number and a lower number. What they are measuring is the blood pressure in your vessels when your ventricles are contracting. So it would be ventricular systole, and then when your lower number would be the ventricular diastole or diastolic number. And that's because we create a lot bigger changes to pressure when the ventricles are contracting and relaxing than when our atria are. So when we really kind of use the terms systole, diastole, a lot of times it's being more referred to as what's taking place with the ventricles, more so than what it is with the atria. But that doesn't take away what we see here as the first of our 5 steps of our cardiac cycle, what we call atrial systole, which if you go to the next page is listed here as atrial contraction. Same thing. What you should do is put systole and then put contraction in parentheses. But this is contraction of the atriomyocytes. So the atria are contracting, the muscle fibers are increasing pressure within the atria, causing causing blood to be ejected from both of the atria into the ventricles. And during atrial systole, we need the ventricles to be relaxed, and that allows for their pressure to be rather low. At the end of atrial systole, that's also the end of ventricular diastole or ventricular relaxation, and each ventricle will contain about 130 mils of blood. We call this volume end diastolic volume, or EDV for short. Literally called that because it's the volume of blood at the end of diastole. Next step is a very short phase, what we call the isovolumetric contraction phase. The beginning of ventricular systole, so the ventricles are starting to contract. The key here is that all valves are closed, and what we see within the ventricles is pressure is going to drastically increase. Okay. The contractions taking place with these cardiac muscle fibers in the ventricles would be what we call an isometric contraction. And we know what isometric contractions are from previous lectures, like doing a wall sit with your skeletal muscles. Isometric contractions can take place with the heart. This is a very, very short amount of time where the heart is squeezing its ventricles, but we haven't gotten that pressure in the ventricles higher than what's in the vessels that it's going to receive. We haven't, created that pressure gradient to eject blood out of the ventricles. So we see no shortening of the muscle in the ventricles, not yet anyway. But when that pressure does go higher in the ventricles than it does in the atria or in the pulmonary trunk, we're going to eject the blood from the ventricles, which we literally just call ventricular ejection. Okay, so occurs when the ventricular pressure exceeds either pulmonary or aortic pressure, depending on if you're talking about the right ventricle or the left ventricle. In other words, doesn't matter. We're talking about blood pressure outside the two ventricles outside the heart, which causes the pulmonary and the aortic valves to open and blood to be ejected from our heart. Okay, we talked about in the last video the size difference between the left ventricle and the right ventricle, and how left ventricle has to pump against higher pressures. Left ventricle can create pressure around 120 millimeters of mercury, whereas the right ventricle, it climbs its pressure internally to only about 25 to 30. Very, very big difference between those two numbers. Our left ventricle ejects about 70 mils of blood into the aorta. Right ventricle pumps about the same amount, which means of that 130 mils that we originated with, we only dumped out a little over half, which means that we've got about 60 mils left, because 130 minus 70 equals 60. We call this the volume, that volume is called the end systolic volume, because it's the volume of blood at the end of systole. At this point, what we need to do is drop pressure drastically so that we can really start to set the stage for returning blood back to our ventricles. So isovolumetric relaxation is stage or step 4, beginning of ventricular diastole, when all valves, that includes not only those semilunar valves, pulmonary and aortic, but also the atrioventricular valves, tricuspid, bicuspid, all of them are closed to drop pressure in the ventricles drastically. What we want is that pressure in the ventricles to eventually drop below atrial pressure, so we can start filling the ventricles back up. And that's our last step, ventricular filling. When ventricular pressure drops below atrial pressure, pressure in the atria, the tricuspid and bicuspid valves will open and blood gets sucked into the ventricles. At the end of this phase, our ventricles are about 3/4 full, and then the rest of the blood enters the ventricles when we start a new atrial systole step. So looking at our illustration here on this next page, pretty complicated figure. We're going to kind of focus in on, the stages or steps down here at the bottom, which are really kind of nice and color coded for us, so we can kind of tell them apart here. When we look at volume, let's just say this is the left ventricle, but right ventricle will be about the same. We're going to kind of wait and talk about heart sounds here in a little bit. We'll talk about pressure, and then we can kind of reference the ECG at the top, but we kind of know all about what's taking place in there. So I'll kind of focus on this segment of our figure and this segment, talking about what's taking place. Now, if you look at the figure, we show 2 atrial contractions, which is kind of misleading. This is, the beginning of the cardiac cycle, and then this is beginning of a new cardiac cycle. Sorry. What they didn't include were, the next 4 steps, because we just are including the beginning of the next cardiac cycle. So atrial contraction or atrial systole, as we had it described on the previous page, represents the pink segment of our figures when the atria are contracting. Now looking at our ventricle is already about 3/4 of the way full, but by the time we end our atrial systole, we have completely filled up our ventricle to its 130 mils, like we talked about on the previous page. Okay. In order for this to take place, what we need is pressure in the atria to be higher than pressure in the ventricles. And if we go to this figure at the top, we have a red line representing aortic pressure, which we'll talk about more here in a second. Gosh, this thing's so sensitive. We've got a green line. The green line represents the left atrial pressure, and the blue line represents aortic left ventricular pressure. Looking here in our pink segment during the atrial systole or atrial contraction phase, it's not a big difference, but the green line is above the blue line. Once again, the green line represents pressure in the atria, in this case, left atrium. And then the blue line represents the pressure in the left ventricle. So a pressure gradient is established higher in the atrium than it is in the ventricle, allowing the blood to leave the atrium and enter the ventricle. And then what we have taking place is the next stage when the ventricles are going to start contracting. But remember, all 4 valves are closed and the ventricle is not going to eject any blood. So like I said, it's a really short stage. We have what's called an isometric contraction taking place. And that's because we're drastically bringing pressure up in our left ventricle to the point where it passes the pressure in the aorta. It doesn't create a giant pressure gradient, but it is high enough that we create a pressure gradient higher in the left ventricle than the aorta, which allows for the valve to open up, allowing blood to leave the left ventricle. We see the same thing, just not showing it in this figure for the right ventricle and the pulmonary trunk, and blood is ejected out of your ventricles. And we don't eject all the blood. We said we only eject about 70 of that original 130 mils. So at the end of that systolic phase, we call this end systolic volume, ESV, it's about 60 mils. So the ventricles aren't completely empty. Then we start to relax. And that's very, very evident here with our blue line going right back down towards the bottom, so that we can get to the point where, once again, we establish a pressure gradient where the left atrial pressure is higher than left ventricle pressure, and pressure can, the flow of blood can start to go back into the ventricles. We suck it back in about 3/4 of the way, and then we start the whole process over again by contracting the atria and totally filling them up. So, a lot going on with one heartbeat, but very, very amazing. Now, for any of this to occur, we have to perform excitation contraction coupling, which is very similar to what we saw in skeletal muscle fibers. This illustration here on this page is kind of a lot like the picture that we showed in skeletal muscle fibers, where we see the same features, sarcolemma on the outside, T-tubes, sarcoplasmic reticulum that stores calcium and will release it when the action potential spreads inward. We see the same segments of a sarcomere, A-band, I-band, M-line, H-zone, etc., etc. Basically, the same things take place. Action potential spreads over the plasma membrane, down the T-tubes. Calcium is released from calcium release channels. Calcium binds to troponin, shift in tropomyosin occurs, cross bridges form, and we go through the power stroke causing the sarcomeres to shorten. Okay. Second wave of calcium binds to troponin, yada, 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 yada. I'm not going to get into it anymore because we already know it. And hopefully, anyway, you know it still from just a couple lectures ago. So just wanted to kind of demonstrate that what's taking place in our heart is pretty dang similar to what's taking place in our skeletal muscles when they contract. Now, I promised we'd talk about those heart sounds that were labeled there on that figure a couple slides ago. We can listen to our heart and other sounds within our body through an act called auscultation, which I mentioned at the very beginning of the semester. This is usually done with a stethoscope. And the sounds of our heartbeat come primarily from blood turbulence that results when our valves are actually closing. During each cardiac cycle, there are 4 heart sounds, which were labeled here on this previous page. Notice we've kind of got some squiggly lines at each of those sounds. S1 and S2 represent the biggest here, and those are the kind of typical 2 that you hear because S3 and S4 are really typically not actually heard. S1 is just short for first sound, S2 is just short for second sound, S3 and S4 third and fourth. Simple as that. And first sound is described as a lub sound. It's a bit longer than the second sound and is caused by blood turbulence associated with closing the atrioventricular valves, those valves between the atria and the ventricles, soon after the ventricles begin contracting, going through ventricular systole. The second sound is a dup sound caused by blood turbulence or associated with closing the SL valves, the semilunar valves, which once again are the aortic and the pulmonary valves between the aorta and the left ventricle and the pulmonary trunk and the right ventricle at the beginning of ventricular diastole. So on the exam, make sure you know closing the AV valves causes the lub sound, closing the SL valves causes the dup sound. And as I mentioned, S3 and S4 are usually not loud enough to be heard, but S3 is due to blood turbulence during rapid ventricular filling, and S4 is due to blood turbulence during atrial systole. Figure on the right just shows kind of common sites for putting the stethoscope to listen to the different sounds, something that we have done, just about any and every time we go to the doctor. Not, anything figure-wise that I'm going to ask you to worry about. Okay, so now we have a pretty good understanding of how our heart pumps and how it ejects blood and all that wonderful stuff. I want to talk about how much blood our heart pumps. So I'll give you a second here because I know there's some stuff you need to write out. Okay, reading through. Although our heart has autorhythmic fibers that enable it to beat independently, its operation is governed by events occurring throughout our body, such things as exercise, when we have a fight or flight, sympathetic nervous system active. Our working cells need elevated levels of oxygen and nutrients, meaning our heart must deliver more blood to those working cells, which means our heart's going to pump more often, and it's going to pump harder to get more blood to those various structures. And three things I want to talk about are stroke volume, heart rate, and cardiac output. Abbreviated SV, short for stroke volume, HR, short for heart rate, and then Q, which doesn't really make a whole lot of sense, but Q is short for cardiac output. Stroke volume is the amount of blood that's ejected from a ventricle per heartbeat, or if you wanted to say per heart stroke, might be an easy way to think of it. So I always tell students, if you think of, your heart like a juice box. I have kids, we do juice boxes all the time. My 2-year-old doesn't quite know, the ramifications of squeezing the juice box, like my 4-year-old does. My 4-year-old does a pretty good job with it, but my 2-year-old grabs that juice box and squeezes it, and inevitably some of that juice spills out on the ground. Okay. When he grabs that juice box and he squeezes that juice out, what falls down on the ground is like stroke volume, the amount of blood that's ejected from the heart per beat. Okay. It is at rest about 0.7 to 1 liters or 70 to 100 mLs, but can rise pretty drastically to 130 mLs or even more during intense exercise. And to calculate it, not something I'm going to ask you to do, and we actually did it earlier in the lecture, it was that 130 in diastolic volume, 130 mLs minus the 60 mLs, and that was the ESV. So going back, it was this number minus that number. So we already kind of talked about stroke volume. Okay, and in fact, if I go back to this illustration, here's a little kind of bracketed off region in our figure that labeled stroke volume 130 minus the 60, so about 70 or 60 or so, somewhere in that neighborhood. Okay. Heart rate is just the number of beats that our heart takes per minute, what you measure when you take a pulse. About 75 beats per minute, or BPM for short, is how you'll see that sometimes listed, but can elevate to around 200 beats per minute during intense exercise. Now, our heart rate our maximal heart rate is a factor of age. The easiest method of kind of getting an idea where your max heart rate should be is just a calculation 220 minus your age. So a lot of you are around 20 years old. So that's why I've put 200 beats per minute. I'm in my 30s, so not quite 200 beats a minute should my maximal heart rate be. You take the number of beats your heart takes per minute, you multiply it by the amount of blood it ejects per heartbeat, you get cardiac output, okay, which is what this calculation is at the bottom. Stroke volume times heart rate, the amount of blood that flows out of our left ventricle or right ventricle. Kind of, we usually measure or are more concerned with left ventricle each minute. So it's the amount of blood per minute that flows out of our heart. It's easy to confuse stroke volume and cardiac output. Stroke volume was per stroke or per heartbeat. Cardiac output is per minute. Okay, we generally measure it using the liters of blood per minute, or liters/min. That unit of measure is usually about 4 to 6 in most healthy people at rest, but can rise quite a bit during intense exercise, which kind of brings me to this page. Not something I'm going to ask you to know. It's something I snagged from my exercise phys textbook to show what being a sedentary individual does to us versus being a very trained individual, getting some exercise, which is very helpful for our cardiovascular system and our overall health. So we show untrained men and women, trained men and women at rest, untrained men and women, and trained men and women at max exercise. Looking at just overall cardiac output, really not much different between the two for either trained or untrained men and untrained and trained women. Pretty much the same, okay? But here's where the difference lies. We pump the same amount of blood per minute, but we don't have to pump our heart as often at rest because our heart can pump out a lot more blood. So it can squeeze out more each time it beats, so it doesn't have to beat as often, okay? If you subtract 22 or so beats per minute, or around 20 beats per minute at rest, you take 20 times 60 times 24, that's a lot of beats that your heart doesn't have to take in a single day. And then you multiply that out by a year, and then by 10 years and 20 years. I mean, being in shape allows your heart to kind of have a little bit longer and better ability to rest. Now, when we exercise, our heart doesn't beat as often, doesn't have to beat as often, because it kind of the same thing, beats and pumps out a lot more blood. Look at the differences in the amount for a trained versus an untrained male, and a untrained versus a trained female, meaning that there's going to be a heck of a lot more blood flowing to those working skeletal muscles, and they're going to get more blood, more nutrients, more ATP. More ATP means more energy for the muscles to contract, and people that are highly trained are going to be more successful athletes. So pretty common sense for the most part, but now you kind of can see why when it comes to cardiovascular and cardiac health, why being physically active and regularly physically active is very, very beneficial. Okay, we're going to talk about things that can affect our stroke volume, and then we'll talk about things that affect heart rate. Reading through, a healthy heart pumps out the blood that enters its chambers during the previous diastole. In other words, a healthy heart's going to pump out what it has in it when it is filled up. In other words, if more blood returns to the heart during diastole, then more blood is ejected during the next systole. At rest, our stroke volume was only about 50 to 60% of our end-diastolic volume, because about 40 to 50% remains in the ventricles after each contraction. Or in other words, we still have about 40 to 50% of our ESV, but that number changes quite a bit, which I won't really get into, as we exercise. When we are bringing more blood back to the heart, we're going to pump more out. There are 3 factors that affect stroke volume. 2 extrinsic, which are related to things outside the heart, and then 1 intrinsic. Okay, the first extrinsic factor is called preload, which relates to something we've kind of seen before with that length tension relationship. Back when we talked about skeletal muscles, we learned that it had everything to do with the number of cross bridges. The more cross bridges, the more force a heart beats. Okay? And so let's just read through and I'll explain what that means. So preload is the amount of stretch placed on the ventricular myocardium before it contracts. An increase in preload or filling pressure results in an increased force of contraction, which means that there's a greater volume being ejected, or in other words, increased stroke volume. So an easy way to write it down, you can just say any increase in preload increases stroke volume. So any increase in preload increases stroke volume. Any decrease in preload decreases stroke volume. So it's a proportional relationship. Preload goes up, stroke volume goes up. Preload goes down, stroke volume goes down. We call this phenomenon the Frank-Starling law because it was first described by a couple scientists, Frank and Starling. It is, as we said, proportional to end diastolic volume, which is the volume of blood at the end of diastole, at the end of rest. Okay. This is affected by the duration of ventricular diastole, but also something we'll get into in our next lecture, what we call venous return, which is exactly what it sounds like, volume of blood that returns to our right ventricle from our venous circulation. Okay. When our heart fills up more, it stretches more, and that stretching gets it to that optimal length, like what we showed with our figure in our muscles lecture, where we maximize the amount of cross bridges that can be formed, and thus more cross bridges mean more force. More force means more stroke volume. So we eject more blood from the heart. Okay. Now, these dotted lines represent the fact that you could see it starting to go down as we stretch out too far, but cardiac muscle fibers, as it says, do not typically operate along the descending portion of our length tension curve, meaning that, we can't see this in a normal situation. The second extrinsic factor is another load. This is called afterload, and it's the pressure our ventricles must exceed to eject blood into the circulation. In other words, this is describing what blood pressure does to your stroke volume. If pressure is high, our ventricles can eject as much blood, causing decreased stroke volume because not enough force can be produced. So what essentially happens is, if we look at this figure again, there's aortic pressure. It's up here instead of down here where it belongs. And so it's harder for us to get this blue line, the pressure inside our left ventricles, up high. And so it just basically makes us operate at a higher and less efficient pressure gradient. And what has to take place is our heart has to beat harder every time. It's really exhausting on the heart and basically wears it out to the point where it can cause the heart to not work very well, causing heart failure and that kind of stuff. So, in other words, afterload is something that has an inverse relationship to stroke volume. Any increase in afterload causes a decrease in stroke volume. So if afterload goes up, preload goes down. Things that cause afterload to go up would be hypertension, which is a fancy word for high blood pressure, and narrowing of your arteries due to atherosclerosis, which is fatty plaque stuff that builds up in the arteries, and that a lot of times causes hypertension. Hypertension, atherosclerosis, a lot of those conditions you see that affect the heart are just related to lifestyle. Proper diet, proper amounts of exercise really can kind of help prevent and treat those conditions. So once again, any increase in afterload causes a decrease in stroke volume, or if you said any decrease in afterload increases stroke volume. Okay, the final factor affecting stroke volume is called contractility. It's an intrinsic factor. And contractility is the amount of force a myocyte can produce based on chemical changes that occur within the tissue itself. Any increase in contractility increases stroke volume. So it's another proportional relationship. Contractility goes up, stroke volume goes up. Contractility goes down, stroke volume goes down. Okay? And that's what we show here on our figure on the right of the next page, with our blue line being the control. Any positive inotropic effect is going to increase contractility, make it beat harder, and harder beat causes stroke volume to go up. Things that cause substances to increase contractility are called positive inotropic agents. Includes stimulating the sympathetic nervous system. This is why when you have that fight or flight response, it might feel like your heart is just like beating out of its chest. Feels like it's beating really hard. That's what it is. It's higher contractility. Hormones like epi and norepi. Obviously, epi, known as adrenaline in a lot of places, or in this country. We feel that adrenaline rush. We know that feeling whenever the fight or flight response is active. And then an increase in calcium that's in our interstitial fluid are all positive. Any substance decreasing contractility would be a negative inotropic agent. What's shown here in a pink line. It includes inhibiting the sympathetic nervous system, acidosis of the blood, and increased potassium levels in the interstitial fluid. So make sure you once again know any increase in contractility increases stroke volume, whereas any decreased contractility decreases stroke volume. And I also would make sure you know that can cause what the positive inotropic agents are, what the negative inotropic agents are, and what that means. Okay, on to regulating heart rate. The other component of our cardiac output is our heart rate, how fast or how often our heart beats. Something that's super important is adjusting our heart rate to control not only our cardiac output, but our blood pressure, which is something that we'll be getting into more in our next lecture. Our SA node, once again, is where we initiated contraction. And as I mentioned earlier, if you left it alone, it would set a constant heart rate of about 100 beats per minute. But due to homeostatic mechanisms, we must adjust heart rate to match tissue requirements under certain conditions, like when we're in an exercising state, and we need to increase the flow of blood to our working skeletal muscles. Two major factors that regulate our heart rate are signals from the autonomic nervous system and certain chemicals. So let's talk about how we affect the heart rate using the nervous system. Thinking back to our lecture, when we introduced sympathetic and parasympathetic, will really help us here. It'll kind of hopefully make a lot of this pretty common sense from stuff we've learned so far this semester. So reading through, nervous regulation of our heart originates in the cardiovascular center of our medulla oblongata. This region of our brainstem receives input from a variety of sensory receptors, which we'll get into just a little bit. Not so much in this lecture, but more so in the next lecture. But we have sensory receptors in our muscles that monitor movements. We also have them in our joints and tendons that are sending signals up into our brain. We've got chemoreceptors monitoring the blood chemistry of things like oxygen, hydrogen ions, carbon dioxide, and baroreceptors, which are receptors that monitor the pressure of our blood vessels, so what we would call blood pressure. Those send signals into our cardiovascular center. We've also got signals coming down from higher brain centers, from places like our cerebral cortex, from our limbic system. Remember, the limbic system is our emotional brain, and how emotions can affect our heart rate, thus also affecting our blood pressure. And then signals from that major regulator of our sympathetic, parasympathetic, our autonomic, being the hypothalamus right up in here. Okay. So region of the brainstem receives a variety of sensory receptors from higher brain centers, such as sensory receptors and from higher brain centers, such as our limbic system and cerebral cortex, then directs appropriate output by either decreasing or increasing frequency of nerve impulses in both sympathetic and parasympathetic branches. And based on what we know, the sympathetic nervous system, which produces a fight or flight response, is going to help increase your heart rate. Okay, whereas the rest and digest response produced by our parasympathetic division is going to decrease heart rate. Okay? Pretty simple, hopefully, for us by now. Okay? Now, sympathetic nervous stimulation, what we're showing off here in this illustration, involves signals coming out to not only our SA node, the AV node as well, but also the ventricular myocardium by way of nerves that we literally call cardiac accelerator nerves. Cardiac accelerator nerves could not be any simpler named because that's what they do. They accelerate the heart because they trigger the release of norepinephrine that binds to beta-1 receptors on the cardiac muscle fibers, increasing SA node depolarization, meaning the SA node fires more often. And that's what we're showing here in this illustration on the next page. Okay. Letter green shows what takes place when the sympathetic nervous system is active compared to the black lines showing in a normal non-sympathetic stimulated situation. More fired action potentials in our SA node. And that's because when that norepinephrine binds to the beta-1 receptors, it activates a second messenger system, domino effect, causing sodium to run into the SA node at a high rate, and the SA node hits threshold a whole bunch, meaning a whole bunch of action potentials are generated. Okay. So not only does the sympathetic nervous system increase our firing of the heart, meaning heart rate goes up. We just talked about it also increases contractility because it increases calcium inflow, which relates to our contractility stuff that we just talked about. So both the sympathetic nervous system activates increases in heart rate and stroke volume. Okay. Whereas the parasympathetic, by way of the vagus nerves that travel and terminate on the SA node, the AV node, and the atrial myocardium, triggering the release of acetylcholine. Acetylcholine is going to basically close those sodium channels, not completely, because we're still going to allow some to flow in because we can't shut the heart off completely. But what happens is we slow the rate of depolarization of the SA node compared to a normal situation shown in black. So this decreases heart rate by decreasing SA node depolarization. We don't see as many vagus nerves innervating the actual ventricular myocardium, so not as many of the ventricular myocytes are innervated, meaning that when the vagus nerves are active, they don't really have as much of an effect on ventricular contractility, and thus really not as big of an effect on stroke volume. Other factors that regulate cardiac output include chemicals. Hormones are a big one, so are various ions. And by now we have a pretty good idea on hormones, hopefully, from our previous lectures, especially epi, norepi that we've talked a lot about, which are always released, as we know, whenever the sympathetic nervous system is active, released from the adrenal medulla, and increases not only heart rate, but also contractility. Same thing we just saw by doing this, binding to the beta-1 receptors, or by causing the release of more calcium, increasing contractility. So epi and norepi increase heart rate and stroke volume. The thyroid hormones T3 and T4 also do the same thing, increase contractility, so stroke volume and heart rate. So epi, norepi, T3, T4, all four of those are going to increase heart rate, stroke volume, and therefore they increase cardiac output. Cations, potassium, calcium, sodium all have a large effect on cardiac function. Elevated levels of potassium or sodium are going to actually decrease heart rate and contractility, which means that heart rate, stroke volume go down whenever potassium or sodium are elevated in our blood, which means cardiac output goes down. whereas elevated levels of interstitial calcium speeds heart rate and increases contractility. So calcium increases heart rate, increases stroke volume, therefore increases cardiac output. Other factors include your age. At kind of newborn age, our resting heart rate's much higher, about 120 beats, and then that gradually declines as we get older. That also affects, like I said, our maximal heart rate, which is why we calculate it as 220 minus our age. Adult females have a higher heart rate than adult males. I've really never researched why that is. Talked about how being physically fit may exhibit a condition called bradycardia. Not a condition that's bad, but is actually a great thing, decreased heart rate. Talked about that with that table that I was showing you when I talked first about cardiac output, stroke volume, and heart rate. Increased body temperature, like during fever or exercise, causes increase in heart rate, whereas a decreased body temperature decreases your heart rate. And a lot of that has to just do with the speed of metabolism and stuff like that, and the need for oxygen and nutrients, but I won't really get into that. Figure here on that last page, or this last page, I guess, just kind of demonstrates the different factors that we've been talking about for the last 10 or so slides that affect cardiac output, the 3 factors that affect stroke volume. In this case, we're showing increased stroke volume. Down at the bottom are the factors that affect heart rate. In this case, showing you increases in heart rate. If we flipped it and we wanted to decrease cardiac output, we would, have to change things out. For decreasing stroke volume, we can decrease preload, we can decrease contractility, we can increase afterload. For heart rate, we can talk about all these different things that decrease heart rate, how the parasympathetic decreases heart rate, etc. But this figure is showing you increased cardiac output, so we will not worry about that. Okay, that wraps us up, so I will shut her down. Thanks, guys.